Advances in machine learning, ML, have produced systems that perform tasks once thought to require uniquely human intelligence. Recognizing faces, translating languages, diagnosing disease, and navigating complex environments. Central to many of these advances are artificial neural networks, ANNs, computational models inspired by biological neural circuits. While ANNs are not literal replicas of the brain, their architecture, training processes, and emergent behaviors show striking parallels to human cognitive mechanisms. Understanding these connections clarifies both how contemporary AI learns and how neuroscience and machine learning can inform one another. Architectural parallels. From neurons to layers at the most basic level, ANNs borrow the neuron metaphor. Biological neurons integrate inputs through dendrites, generate nonlinear responses at the soma, and transmit spikes via axons. Artificial neurons similarly aggregate weighted inputs, apply a nonlinear activation function, and pass results forward. Collections of artificial neurons are organized into layers, input, hidden, and output, mirroring the hierarchical structure found in many brain regions where information flows through successive processing stages. For example, from primary sensory areas to higher-order association cortices. Convolutional neural networks, CNNs, widely used for image processing, explicitly echo the visual cortex's organization. CNNs use localized receptive fields, weight sharing, and hierarchical feature extraction, principles analogous to how the mammalian visual system detects edges, textures, and complex shapes through progressively abstract representations. Recurrent neural networks, RNNs, and architectures with feedback loops capture temporal dynamics and persistence of information, similar to recurrent connectivity in cortical and subcortical circuits that underlies working memory and sequence processing. Learning mechanisms, optimization and synaptic plasticity. Learning in ANNs typically occurs via gradient-based optimization, e.g., backpropagation, which systematically adjusts connection weights to minimize error on a task. Biological learning is driven by synaptic plasticity, changes in synaptic strength in response to correlated activity patterns, Hebbian learning, neuromodulation, dopamine signals for reward, and homeostatic mechanisms. While backpropagation is not a biologically exact process, conceptually it plays a role analogous to synaptic adjustment. Both modify connections to improve future performance. Recent research has built bridges between these domains. Variants of local learning rules, spike timing-dependent plasticity, STDP, and biologically plausible approximations to backpropagation show that neural-style learning can arise from local, activity-dependent synaptic updates. Reinforcement learning in AI, which pairs trial and error updates with reward signals, maps naturally onto dopaminergic reward prediction signals in the brain. These convergences suggest that, though different in implementation, both artificial and biological systems optimize connection strengths based on experience-driven feedback. Representation and abstraction, from sensation to concept. A critical capability of both brains and ANNs is building representations, internal codes that capture regularities in sensory input and support flexible behavior. In deep networks, lower layers tend to represent simple, local features, e.g., edges, while deeper layers encode increasingly abstract, task-relevant patterns, e.g., object identity. Neurons in the brain similarly exhibit a gradient from low-level sensory tuning to high-level, multimodal representations in association cortex. Moreover, both systems demonstrate compositionality and disentangling, learning to separate independent factors of variation, such as shape versus color, supports generalization to new combinations. Techniques like unsupervised representation learning and self-supervised learning in ML replicate the brain's ability to extract statistical structure from raw sensory streams, reducing dependence on labeled examples and enabling transfer across tasks. Perception and predictive processing. Modern theories of brain function emphasize prediction. The cortex continually generates expectations about incoming input and updates them based on prediction errors. This predictive coding framework aligns with many ML approaches that optimize models to anticipate future inputs or reconstruct missing data, e.g., autoencoders, sequence models. Neural networks trained on prediction tasks often develop internal representations and dynamics that resemble those observed in neural recordings during perceptual tasks, reinforcing the idea that prediction is a core computational principle shared across biological and artificial systems. Robustness, generalization, and failure modes. Despite these parallels, differences matter. Biological brains are robust to noise, adaptable with few examples, and capable of reasoning in open-ended environments. ANS can be brittle, susceptible to adversarial perturbations, prone to overfitting, and dependent on large labeled datasets. Some of these gaps reflect architectural and training differences. Brains exploit massive recurrent connectivity, neuromodulation, lifelong learning, and embodied interaction with the environment, factors still only partially realized in ML systems. Understanding failure modes in both domains is informative. For instance, adversarial examples reveal that networks may rely on statistical regularities that diverge from human perceptual priors. Neuroscience studies about illusions and biases similarly show that human perception depends on priors and heuristics, producing both strengths and systematic errors. Comparative study of these errors can guide the design of more robust, human-aligned AI. Reciprocal benefits. What AI and neuroscience teach each other? The exchange between ML and neuroscience is bidirectional. Neuroscience has inspired architectures and algorithms, convolution, recurrence, attention, while ML offers quantitative tools for modeling complex neural data and testing mechanistic hypotheses at scale. Deep networks trained on ecologically relevant tasks serve as computational probes, whose internal representations can be compared to brain activity, helping to identify principles of sensory coding and decision-making. Conversely, incorporating biological mechanisms, sparse coding, dendritic computation, neuromodulatory gating, into ML holds promise for more efficient, adaptable AI. Ethical and philosophical considerations. Drawing analogies between machine learning and brain function invites reflection about agency, interpretation, and responsibility. While ANS may mimic aspects of cognition, they lack subjective experience, intrinsic motivations, and the rich embodied context that shapes human minds. Overextending metaphors risks anthropomorphizing systems and misattributing capabilities. Responsible development requires clarity about where parallels are explanatory versus where they are merely useful metaphors. In conclusion, machine learning and neuroscience converge on shared computational themes: hierarchical representation, experience-dependent synaptic modification, predictive processing, and the extraction of abstract structure from sensory streams. Artificial neural networks capture many formal aspects of brain computation, providing powerful models for prediction and control while also exposing gaps in robustness, flexibility, and embodiment. Continued dialogue between the fields promises richer theories of intelligence and practical advances, AI systems informed by biology, and neuroscientific inquiry sharpened by scalable computational models. Understanding both the parallels and the limits of the comparison is essential as we develop technologies that increasingly mirror the functions of the human brain.